Hello, good evening. My name is Lamborn and I'm going to talk to you today about taking part in the UK Pollinator Monitoring Scheme. Now, I've got to give credit where credit's due, first of all. And this talk was, originally the bulk of this talk was written by my colleague Richard Dawson, and Richard and I presented it at the Big Buzz conference in Cumbria in last September. So firstly, I'd just like to thank him for putting all the hard work that he did putting this talk together. And I'd also like to thank the organisers for inviting me and for everybody to coming this evening. Thank you very much. So, I'm just going to introduce you a little bit to the team. The POMS team. POMS was started by Dr. Claire Carville at the Centre for Ecology and Hydrology in Wallingford, Oxford. And it's a very small team, actually, of dedicated ecologists who work full-time on the team. The rest of us are freelance ecologists. Richard and I are both volunteer mentors, which means we look after the volunteers that we have working on the scheme. And Richard started with POMS as a volunteer in 2008 when he adopted a square, one of our survey squares in the Peak District. And I was the volunteer mentor I'm the volunteer mentor for the northern squares. And so I went down there to train him. And he loved the job so much that when the role came up in North Wales, he moved over to North Wales to do the same job that I do with the mentoring. He's a specialist in solitary bees and wasps, and he also has a background in design and creative engagement, and tries to get people and the public involved in positive environmental projects like POMS. He also runs an entomology accessories company, if you happen to want any little accessories for entomology. And both of us have actually been working together this winter to identify bees collected from the POMS work last year. We're together with 3 other entomologists who are looking at the hoverflies. They're identifying hoverflies. For myself, I started in pollination ecology in the late 90s when my project supervisor at my undergraduate university suggested that I studied the insects of wild carrot, which I went on to do for my final year thesis, and then I published that work. I then moved to Exeter and I did a PhD at Exeter on plant reproduction. And then after that, I popped over to Greece, and I worked in Greece for a few years in the Aegean on a European project that was looking at pollinator loss. And we were specifically testing the methods of how we can actually measure insect diversity and abundance. So we were testing which methods to use. I specialise in bee identification, recording and plant reproductive ecology, and I've been working on a number of projects recently. Like last year, I worked on a project for the University of Leeds, the University of Oxford and the Royal Botanic Gardens at Kew. And what they're doing there is they were looking at bee nutrition, and I had to go out and collect pollen from about 100 or more species of plants in the north so that they could have a northern replicate, and they could look at that for bee nutrition. And I've also been doing some work for CEH on the POMS project, and they've also some work on their agri-environmental schemes. So that's me. So what's POMS? So POMS stands for the UK Pollinator Monitoring Scheme, or POMS for short. So when we talk about pollinators, we're just talking about flower-visiting insects that assist in plant reproduction. We're not qualifying whether they're any good at being pollinators. We're just looking at them as flower-visiting insects. POMS is the world's first monitoring scheme that's generating systematic data on the abundance of bees and hoverflies and other insects at a national scale. So there are local recording schemes and monitoring schemes, obviously local projects going on, And there are also a lot of recording schemes, but they're looking at individual things. So you've got the Bumblebee Conservation Trust, the Butterfly Conservation Trust, you've got the Hoverfly Recording Society, you've got the Bees, Wasps and Ants Recording Society. What POMS is doing is bringing that together under a sort of an umbrella of pollinating insects. So we're combining the long-term records from these recording societies who are partners, and we're linking them with 2 new large-scale surveys that we're going to use to track, or we are using to track insect populations across the UK. They are the systematic 1-kilometre surveys and the flower insect time counts. I'm not going to talk to you much about the 1-kilometre surveys. I'm just going to talk about it briefly because there aren't any squares in the north. I don't know what they think they're playing at, but they haven't put any squares in the north. And so what I really want to do today is to talk to you about the flower insect time counts and try to get you out doing them, basically. So firstly, why do we need POMS? Well, I know you've had Dave Goulson in recently, so if you saw his talk, then if it's anything like the one in Cumbria that I went to, he was talking about the insect apocalypse and insect declines. And studies looking at insect declines in both Europe and the UK have tended to show these alarming reductions in both species distributions, how they're distributed across the country, and also in diversity, so whether we're losing species, and also when they've been studied, also in abundance as well. But one of the things, most of the data that we have on these species declines are from dedicated volunteer recorders, basically, who've contributed to the recording societies. But it's often difficult to assess what's happening with the pollinator populations due to a lack of data. The European Red Data List actually has quite a lot where it just says data deficient, data deficient. We don't know. We don't know what's happening with these populations. So I just wanted to sort of at a bit of an offside, really. I watched a really good talk on YouTube recently. It was by Doug Tallamy, and it's called A Pollinator's Best Hope. I'm just going to read what I've written about it. So it's a new approach to pollinator habitats that starts in your yard. And it's about an hour and a half long, but it's well worth it. And he thinks that we made 3 missteps in the nature conservation movement. The first of those missteps was that we sort of think of nature as a bit optional. Yeah, it's something we need to save nature so that our children, future generations can enjoy it. But what we don't recognise when we talk about it like that is that it's not about enjoyment. We are fundamentally, we need nature to survive. So not just saving nature for future generations, we need to save nature so that we can have future generations. The second assumption that we've sort of made with nature is that we can't coexist with it, that we've got to have it in these isolated pockets, national parks, local nature reserves, things like that, that we can't actually live together with it. What that does is it means that this is ecologically significant, but your back garden isn't ecological. Well, of course it is ecologically significant. Your back gardens can be really ecologically significant for pollinators. And then the third thing is that we've sort of left the stewardship of the earth to a few specialists, really, ecologists, conservationists, natural historians, scientists. So rather than seeing the Earth's stewardship as something that belongs to all of us and we're all responsible for, what we've tended to do is, well, they're looking after it, it's right, they'll look after it over there, and we don't actually do anything for ourselves. But actually, the Earth's stewardship, we're all responsible for it because we're all affected by the quality of our ecosystems. So rather than trying to squish our nature into little pockets far away, we've got to try and fill in the gaps that we've caused by agricultural intensification and urbanisation. We've got to try and fill in those gaps with plants and insects, and then we've got to try and understand them. So one of the biggest and most important contributions that POMS brings to the table is that it gives you the opportunity, everybody, it gives all of us the opportunity to go out and actually look at pollinators, study them, and send in your results, and we can all take a responsibility. Now, I could make this talk about gardening and what to do in your garden, but actually there's a lot of books and things on that subject already. What I want you to do is to go out there and just look at what's there first of all. So, the impact of POMs so far. It's contributed to several publications in the UK. It's part of the monitoring, the National Pollinator Monitoring Plan, or the Action Plan from the Department for Environmental, Food and Rural Affairs. And the methods have also been used or are informing stuff going on in the US national programme, and also in a proposal called Spring for the EU Pollinator Monitoring Scheme. So people are enjoying this methodology and they're wanting to use it. Fit counts are currently in use in Ireland, Jersey, Cyprus, Chile, Brazil and Argentina. So I'm just going to go back a minute and talk a little bit about our 1 kilometre survey squares. They are a network of 95 1 kilometre squares. They are co-located across most of their range with the National Plant Monitoring Scheme, which means that at some point in the future we're going to be able to link up the data that we get. And they're designed to detect broad changes in insect abundance and more specifically investigate species richness and distribution of bees and hoverflies. These 1 kilometre surveys are volunteer-led, so we try to get a volunteer to adopt a square, and then when they've adopted that square, they go out once a month between May and August. That's 4 site visits a year, and they set out 5 pan traps. Here. So they set out 5 pan trap stations. This would be a pan trap station with 3 little pans of soapy water. They're on a post as well, there's other things. And they set out 5 of these little stations across the 1 kilometre square. They leave them out for 6 hours. And then they go back and collect them afterwards. Now, the pans are filled with a soapy water. The soap breaks the surface tension of the water. The insects go in and they can't get back out again, which does mean that this is lethal sampling, and it's not for everybody, and that's absolutely fine. But the volunteers that are working on it, it gives us the opportunity to get accurate species records so that we can see whether things are declining. So what they do is they're also asked to conduct 2 flower insect time counts or fit counts, and then they go out and collect the pans back in and pop them in little things like this, and then they send them down to CEH. And these are the squares. As you can see, there's a big gap in the middle. So 106 is Massam and 118 is near Jedburgh. So in the Scottish borders. So yes, we don't have any around here, which is awful because I have loads of people who'd be interested in doing it, but there we go. I look after the top 7 squares. I go down to 180, which is in Worksop, and I look after the volunteers who work on those squares. All the equipment's provided by POMs, the initial site visit. We go out with you and we give you full training. And then we mentor you through the summer if you need any help at all, if you're worried about anything that you're doing. So if you happen to know anybody who you think might enjoy this, it is strenuous, but it can be really, really enjoyable, then please pass on the email address to them. So this is the main bulk of what I want to talk to you about today. Flower insect time counts. How can you help? Every single one of you here can help. These flower insect timed counts or fit counts are, well, the beauty of the fit counts really is that they can be done by anybody with 10 minutes to spare and some flowers. So they don't require any strenuous activity, so people with limited mobility or energy can get involved, and they're a really great activity to get your kids involved and looking at nature. The flower insect time counts collect data on the abundance of insects, of flower visitors. So to put it very simply, all we need to do is sit down and count the insects that land on our target flowers in a 50 by 50 centimetre patch during a 10-minute period. And it sounds simple enough, really, and that's all I'm going to do is convince you today that it is really simple, and we should all get out and do it and spend your summer enjoying the sunshine, counting bees and hoverflies and insects like I do. So, we do need to do a bit of preparation. So the first thing is to set up an account on the POMS website under the menu. You can do that pretty straightforward, and then you can get involved. You can press the get involved for info and resources. There are videos, there are printables, there are downloadable species guides for flowers and insects. And then if you happen to use a smartphone, you can also download the app on the Play Store or the Apple equivalent. And I think, yes, and you can use the app instead, which is quite useful, because then you don't have to print out a fit count recording form. Oh, I had one here, there you go, for example. So basically, you would print one of these out. If you want to go old school and have it on paper, that's absolutely fine. And basically, that's what you will need to do your recording, or if you have your phone, you can just do it all on your phone. Then the last bit of prep that you need to do is to make a quadrat. Now, quadrats are square, 50 by 50, and all they're going to do is mark out your patch so you don't get distracted by the flowers and insects all around it. So they can be made from string, twine, wool, knotted at the corners. This one's made out of gaffer tape. And if you're into metal and woodwork, you could make a permanent one if you wanted to. But I've just got one in with gaffer tape, and I can pop that in my pocket. That's really easy to use. String's probably easier. The only requirement is that they're 50 by 50 and that they're square. So now we can move on to doing the count. We need to check that the weather's good. It needs to be dry. It needs to be over 13 degrees if it's sunny, and over 15 if it's cloudy. I have done it in less, because it can get a bit cold sometimes. In spring, but yeah, at this point, I'm actually going to say, if you don't see anything when you're doing a fit count, that's still just as important. In fact, that's one of the beauties of the fit count method, is that we get to record when you don't see something, and something not being there is generally not recorded. We record when something is there. Oh, I saw a bumblebee, I'll put it on iRecord, or I'll send it off and tell Bee Wars about it, or something like that. But we don't say, oh, I didn't see anything today. So having this like timed point that where you're looking at flowers and you don't see anything, that's really important. It's really good information for us to get. So you've got your weather, that's absolutely fine, and you need to choose a target flower. We have a list. There is a target flower list. If you can't find a target flower, though, you can use any flower. And if you're not quite sure, take a photo of it and you can upload that at the same time that you upload your results. There's a flower guide online that will also give you an idea of when the plant's in flower. So if you want to do a fit count in May, you can go and have a look and say, oh, these flowers are in May, and you can do that. Or you can choose from the options in the app, or if you're not choosing a target flower, you use the other. Then you need to mark out your patch. So you're going to put your quadrat down on your patch, and the patch can be up on a hedge, on a tree, or anything like that. And then you need to count the flowers in the patch by flower unit type. So here we've got 12 buttercups. And what I mean by flower unit type, it's basically a way of making sure that you're not sitting counting individual flowers of things like heather or something like that, yeah. So buttercups, there would be 12 buttercups in that quadrat. And then in the other quadrat, you can see you've got the 1 dandelion and you've got the 10 daisies. Now, we're not counting the dandelion because we're not studying the dandelion. We're just going to study the daisies. There are 10 daisies, and they're counted as flower heads because we wouldn't expect you to count all of the little flowers in a daisy. It's a composite. So these are the flower unit types. You've got individual flowers, buttercup, hawthorn, bramble, those type of flowers. You've got flower heads, dandelion, daisies, clover. We include ivy in that one as well. They're often really good for insects, so they're quite, they can be an enjoyable fit count to do. And you've got flower umbels, cow parsley, wild carrot, something like that. They look like sort of inside-out umbrellas. We don't expect you to count every flower, so just the unit is the umbel. So if you're looking at a plant, you can count the umbels. And then with a flower spike, it's the same thing. Heather, lavender, white dead nettle, you just count the spike. So it saves you a bit of time with the counting. Then you need to start your 10-minute timer, count all the insects that visit any of your target flowers within your patch during the 10 minutes. You don't count anything that's already there. So if you've got a load of pollen beetles on there, you don't need to count them. And if you can, if you do see an insect fly in, land on your flower, fly out, land on a flower just outside the patch and fly back in again, we'd rather not have duplicates if you see it, but obviously you can't see everything. And then we want you to identify each insect visitor to a broad group. There is a group guide, an insect group guide that's downloadable from the website. There's also one on the app. And these are the insects, the broad groups that we want you to identify to. So bumblebees, honeybees, solitary bees, wasps, hoverflies, other flies, butterflies and moths, beetles, small insects, and other insects. Now you can see how everybody can get involved with this. And then you tally each insect on your recording form. There are boxes, or if you're using the app, just tap. And then you fill in the location details so you can pick them on the website so that you don't have to go and find the grid reference, it will find it for you. And this, if there's a map on there, you need to put what the weather conditions are, and then obviously make sure that it's linked to your name. And then you input your results and on the website, or you upload directly from the app. So the app's actually a lot easier to use in some ways, but I appreciate that some people might prefer to use the website. That's it. Broad groups. Some people don't want to do this because they are worried about these broad groups. I don't know, I can't tell the difference between a honeybee and a solitary bee. So I'm just going to show you how easy it is, really. It's a moth. No, it's a butterfly. It's a butterflies and moths, yes. That's the insect group for them. So we tick them off. I think everybody knows what beetles look like, do they? Don't they? Do they? I hope so. So that's 2. This one's, you're going with size, yeah. This one, if anything under 3 males, is a small insect, so that gets put in that one. This group is just an amalgamation of everything that doesn't fit in any of the other groups. So you've got lacewings and sawflies and shield bugs and crickets and ants and things like that. They will be on your flowers, no doubt. And this insect group is also really good because if you don't know what you're looking at, put it in other insects. Yeah, so if you're not sure whether you've seen a honeybee or a solitary bee, put it in other insects. We've still got the data then. Then, does everybody know what this is? Talking to a Natural History Society, maybe you all know what this is. Okay, so most of you will recognise the hairs. It's hairy, it's rotund, like myself. It's often they collect pollen on their legs, and you might not have noticed that they have a bent antenna, but that's a bumblebee, okay? And while I was here, I just thought I would show you some pictures of bumblebees in the northeast that you might see when you were doing your fit counts, and how to identify them. Now, POMs, we're not interested in a species level identification, but the more you look at insects, the more you want to identify them to species. So we've got Bombus terrestris, which is the buff-tailed bumblebees, the first picture. The queens come out early in the spring. In the south, when I was at Exeter, we used to see them in January. They'd have overwintering colonies. But up here, they're one of the first ones out. They're chunky bees, the queens. And you can tell them by their buff tail. And now, they have this one stripe on the thorax, and then they have one on the abdomen. And the next one, the Bombus lucorum in the middle, also has the same patterning, but it has a white tail. So now we know that the Bombus lucorum group is actually an aggregate of species. There are several species there that are certainly in the workers and in some of the males, and sometimes in the queens. You can't, they're morphologically so indistinguishable, you can't tell them apart. So they're called the Bombus lucorum ag group. And so if you ever see something like that, it just tends to be all lumped together because you need to do DNA analysis to tell what you've got. So the patterning on Bombus lucorum is very similar to Bombus terrestris, and actually with the workers, they're quite difficult to tell apart as well. But over to the other side, we've got Bombus hortorum, which looks very similar, but if you look in the middle, you'll see that it has 2 bands on the thorax, and one, the one on the abdomen is actually closer to the thorax, and what that means is it ends up looking like it's got a big belt on. Bombus hortorum is also known as the garden bumblebee, and they're quite large bees, but they are quite common. You probably see quite a lot of them. And in contrast, Bombus pretorum in the middle at the bottom is, they can produce some of the smallest worker bees going. They can be really, really tiny and a lot smaller than some of the solitary bees. So Bombus pretorum has the same collar, yellow collar. It has an abdominal band that sometimes in the workers is reduced or gone completely, but it has a red tail. So it has a little pinky red tail. And then finally in the corner is probably the most common bumblebee in the country, which is Bombus pascorum, or the common carder bee, and they're sort of ginger bees. Quite straightforward. With bees, you always get cuckoos. And these are 2 examples of cuckoo bees. I wasn't sure whether this was Bombus bohemicus or Bombus vestalis when I put it, so I just put spur, because obviously if I had it in a jar, I might be a little bit more inclined to look more closely and see what it was. But as it is, it's a northern species. Bohemicus tends to be found more in the north. The cuckoo bees they don't have a nest of their own. They steal the nest of another bee. So what they tend to do is they wait until the queen has established her nest of a social bee, of the first ones that I showed you, these ones. They wait till these queens have established their nest and got quite a few workers, and then they go in and they either kill or subdue the queen and lay their own eggs, and then get the workers, well, the workers will provision those eggs and the larvae as their own. So they don't have workers of their own. They steal the host's workers. They often mimic their hosts in terms of what they look like. The rupestris is a cuckoo on Bombus lapidarius, and the other one, whether it's vestalis or bohemicus, is a cuckoo on terrestris or lucorum. So they're often bigger, they often have darker wings, and they won't be collecting pollen. So if you do happen to see one in your patch, they won't have pollen on their legs because they don't need to, because the workers of the host are doing all the work for them. It's cuckoos for you. In late summer, there's usually lots of male cuckoos hanging around on thistles, so they're always good to look out for, if you happen to pick that as a target flower. So that's our bumblebees. Though things are never easy, are they? There's always going to be a few mimics, and this one here is a fly mimic of Bombus lapidarius. So Bombus lapidarius is the bee, this was called the red-tailed bumblebee, and Volucella bombilians, I think it's pronounced, is a fly. So how can we tell them apart? Well, flies have short, stubby antennae. They have big eyes that take over most of the head. They only have 2 wings, which you might not be able to see very clearly on this photo, but the bee is folding her wings back and has there's a set of wings underneath, and you can see that the fly's actually holding its wings out. So that's another way of being able to tell them apart. Otherwise, they do look quite similar. And we'll move on. They also can be quite, they can be mimics. Now I've told you a little bit about the fly on the previous slide, you might be able to see that we're also looking at a fly here. We've got short stubby antennae, we've got big eyes, we've got stripy abdomen. There's only 2 wings rather than 4 wings. And also, if you look at the activity of an insect, you can often see what it is. So I was out on a cycle ride with my friend and her daughter, and her daughter said, "Oh, it's a wasp, it's a wasp, it's a wasp." And I said, "No, it's not, it's a hoverfly." She said, "How can you tell that?" And I said, "Because it's doing this in front of your eyes. It's quite obvious." She was like, "Oh." I said, "It won't sting you, you're okay." And she calmed down. So she's 9, so she couldn't tell the difference between a wasp and a hoverfly. So hopefully I taught her something, and we can move on from that. So yeah, this is the hoverflies. So they do tend to, once you get your eye in, you'll be able to see these. I'm sure a lot of you already know this. So that ticks off the hoverflies. Now, some of the hoverflies are actually mimicking these, which are probably our most well-known bee. And these are probably one of the ones that people get hung up on. How can I tell the difference between a honeybee and a solitary bee? So, well, to tell it apart from a fly, we've got longer antennae. It's collecting pollen. It's got pollen on its hind legs. So, and again, looking at the wings, you can see on this one actually quite clearly, the one flying, that it's got the 4 wings. How do you tell it apart from a solitary bee? It's got dangly legs. When it flies, it dangles its legs. And those legs are actually quite sort of flat, sort of shaped a bit sort of, well, I don't know, square, I suppose, that's how I would describe them, and they dangle down. They're often covered in balls of pollen, so that's one way of recognising that you've got a honeybee. Their shape is also, their abdomens are sort of quite bullet-shaped. If you were to look a bit more closely, you'd see that their eyeballs were hairy, but who's going to look that close? And that they have the cell margin, the marginal cell of the main wing is a sort of banana shape. But yeah, you probably won't get to see that up close on doing a fit count. So those are the ways that you can tell a honeybee. And then we've got these. So they've got the straight antenna, and they're reasonably long antennae, so that gives us an indication that it's not a fly. The eyes are at the side of the head. They've also, if you look at the hind legs, they've got hairs there to collect pollen. Obviously, these are female solitary bees, and they've got 4 wings, and as you can see in rest, they're sort of laying their wings behind them as bees do. So I just wanted to show you a little bit more about solitary bees. You may already know this, but on the legs, they tend to have dry pollen. So it doesn't look like a ball of pollen like a honeybee does. It's more dry and it sticks to the hairs. The females, obviously these are female free-living species. The females tend to be quite hairy if they collect pollen on their legs, or as you can see with Megachile centuncularis, they actually collect pollen on the underside of the abdomen. Now, just to go back to the Andrena hemorrhoa, a very common species, you'll often see them on dandelions. They have a really foxy red thorax and a red tail. I believe the hemorrhoa stands for red tail or something along those sort of lines. Red bum, maybe, I don't know. And then the Megachile, so Megachile have they collect the pollen on the abdomen. They tend to come out a little bit later on in the year. They're a summer species. And they're also known as leaf cutters. You might notice if you've got some circles of leaves removed from your roses, they quite like them and they use them to line their nests. Now, being solitary bees, they nest in different ways. So the Andrina will be nesting in the ground. She will make a tunnel in the ground and then lay an egg in there and give it a little bit of pollen, and then she won't ever see her offspring. They'll come out the following year. And with the Megachile, they'll often nest in your trap nest, or as they're known nowadays as bee hotels, which is your bamboo nests that people put out in their gardens for Megachile and for Osmia species and other species. So if you happen to see a bee with a lot of pollen on its legs, or you happen to see one with a pollen on its abdomen, you know that they are solitary bees. But you do have a few that creep in that are trying to trick you. So this Hylaeus species at the bottom is they actually swallow their pollen and then they regurgitate it back at their nest. So they don't need to be very hairy, and they tend to look quite wasp-like, I suppose, in terms of not having a lot of hair. These ones are distinguishable, and they're actually known as, their common name is yellow-faced bees. So this is a female. The males have even more yellow on their faces. So if you happen to spot something like that, that's also a solitary bee. So one of the other ways that you can identify solitary bees, or at least get an idea that you're looking at a solitary bee, is by how they're foraging. So this is a breakdown of foraging strategy by bee species in the UK that my colleague Stuart Roberts wrote for the Bee's Knees talk that he did for Bee Wars, I believe, in 2016. And on the sides it says oligolectic, polylactic and brood parasites as the three strategies. So oligolectic, what does that mean? It means specialist. So oligolectic bees are bees that are specialist on a few small taxa of flowers, like the Caletes hederae in the corner here, which is also known as the ivy bee, which gives you a little bit of an idea of what that forage is on. And it tends to use ivy pollen to provision its young. It's a really good one to use as ivy is a very good one to use later on in the year. It's sort of September, late September, October time. And Caletes hederae is actually a bee that's doing quite well. It's been expanding its range, and we've started seeing it. I think in 2017 I saw it in Saltburn. I'm from Middlesbrough. I live in Middlesbrough, not from there. And I saw it in Saltburn, and I was sort of waiting for it to creep up the coast, because they tend to have a sort of range expansion when they go up the coast and then they come in from there. And they visit ivy, they collect pollen, the females collect pollen from ivy, so ivy's pretty ubiquitous across the UK, so we see quite a lot of Caletes hederae heading up. I think they've been found in Scotland now, so... And it only entered the UK in the early 200s, 2001. I think it came into the UK. So it's had about 22 years in the country, and it's coming further and further north as it goes. So that's one that's doing quite well. So it's a specialist. There's also, you might spot, if you do heather on any of the moorlands, you might spot its cousin, the heather bee, and that is Caletes succinctus, which looks very similar. How do you tell it apart from a honeybee? You look at the legs. The legs will be hairier and not as flat like a honeybee, and these stripes are quite distinct, a lot more distinct, I think. So then you've got generalist bees. They're not going to help you out much by what they're foraging on, because they visit everything. That's what polylactic means. And then we have brood parasites, or cuckoos. And in the corner here, I've just popped an example of a Coleoxys species. And they're called sharp-tailed bees. You can see why. And they actually parasitise the Megachile family. From the previous slide, these ones here, Megachile genus, sorry. And what they do is they wait until the host bee, the megachile, has gone off to go and collect more leaves or more pollen, and then they sneak into the nest. They use that sharp tail to push their eggs into the leaf, around the leaf that has been wrapped around the cell. What the leaf cutter bee, the megachile, does, it lines its cell with the leaves, so it just pops an egg in there, and then that egg will come out and it will eat the pollen and probably the larvae as well of the host bee, or the egg of the host bee, depending on whether its timing's okay. Parasites, brood parasites, cuckoos, they don't need to collect pollen because they're letting the host do all the work again. So what they tend to do is they tend to be a little bit less hairy, and there are some species of cuckoos that are very wasp-like. So Nomada generally. I learnt how to say the names of bees when I lived in Greece, and my Greek professor was so everybody else says nomada, but I was told I had to say nomada. Yeah, she was very prescriptive with me. So I do say things differently to everybody else, but as long as you understand. So, Nomada are also cuckoo bees, and they can look quite wasp-like. They have a lot of red and yellow patterning on them, but I couldn't find any decent photos that I had taken, so I just put up the coleoxys there. So those are different ways to give you an idea of whether you've got a solitary bee. These are some really they're not really common. 2 of them are really common, and 1 of them's not very common at all. Andrena cineraria is this beautiful ashy mining bee. It's black and grey, quite distinctive if you happen to see that. It's quite common in the spring. Andrena fulva is this lovely gingery bee. That's also quite common. You might see them nesting. You might actually see some of their nests. They like to nest around tree roots and things like that. And then right at the end here is not such a common bee at all. It's called Andrena ruficus, and it's known as the northern mining bee. And you might see this if you went to a nature reserve early in the spring. I see it in Stockton from mid-February till late March. So it isn't something that you might see if you were doing a fit count, because they tend to be more in the summer, April, May, June, July and August. But yeah, Andrena ruficus, it's rare across the country, but it's one of those bees that seems to like the cold, and it will be out in Scotland in sort of June time. But as I said, I've found in Teesside that it's the first bee that I see. And Andrena ruficus is an oligolect. It's a specialist on willow pollen. So if you happen to have a pussy willow tree anywhere nearby, they love it. There's a lot of bees that love willow pollen, actually. So just if you ever happen to be planting something, or farmers who are get them to put in a pussy willow, they're really great for bees in early spring. So that gives us our solitary bees. I think everybody can work this one out. Okay, we've got this small stubby antenna, big eyes, pair of wings. They look like flies, so they would be classed as other flies. And then finally we've got wasps. So a lot of wasps have these big boxy sort of heads. They don't tend to be very hairy. They won't be collecting pollen. And their activity on flowers will tend to be drinking nectar or actually looking for prey. So they'll be looking for something to eat in terms of another insect, often. So this is your wasps. So basically, we've done them all. If you're unsure which group to put your flower visitor in, use other insects. It's as simple as that. And this is really good because you can get kids looking. Your first time you do it, if you're not familiar with insects, you might be putting most of the stuff in other insects. But when you start looking, when you actually start looking in this tiny world going on around you, then you can start to see the differences. And I must admit, from my own experience, I found it absolutely brilliant for my mental health to just get outside and start spending a bit of time in nature. So, your data, what do we do with it? Well, first of all, I just wanted to show you this, which is how fit counts have been growing in terms of how many we're doing each year. 2017 was the pilot year, and up to 2022, where not all the data's there. But we've had over nearly 40,000 individual insects counted. We've got over 800 participants, and we're going from strength to strength. And obviously, the lockdown effect, people could still do fit counts in their gardens, so people were getting out and doing that, which was fabulous to see. If you happen to go on the website, you will start to see how you can actually look at what's been visiting. So on BlackBerry, you've got we've got a proportion of nearly a quarter of the insects were small insects, a fifth bumblebees, and then you've also got hoverflies as well that were interested in brambles. Buttercup, on the other hand, small insects seem to like buttercup and other flies. And then white dead nettle, actually the shape of the flower tells us that we'll probably be expecting something with a longer tongue, and that would be your bumblebees. So what you can do is once you've put in your data, you can start to download your data. So if you happen to be running a project, we haven't actually set it up yet where you can umbrella a project under one thing so that lots of different people can contribute to one project, and then the organiser of that project can download all the results at the moment. But this is something that we are planning on doing. But what you can do is, if people put in their own data, then they can download the report as a CSV file, and then they can hand it all to the organiser, so that if you do want to be looking at your local park or gardens, or if you've got a project that's studying insects, you can use this technique and method. And then you can also explore your fit count results for the area. Now, this was 2022, what we've had near Newcastle. So I thought I'd download to show you that. That actually goes all the way over to Carlisle and down to where I am in Middlesbrough. And you can see, basically, we need to be doing a few more. It'd be lovely to get people out doing more. And then I'd just like to thank everybody that has contributed to this talk and contributes to POMS. We've got the funders, the partners, the survey teams, the landowners that let us do our surveys on their land, the photographers that have given me photos for this talk, and all the volunteers. That's just a list of people. And then that's just I'll just leave that up there for you to just a sort of rundown, really. Wait for good weather, do as many fit counts as you like, get the app if you want, contact us if you're interested in the surveys, and you can keep track of POMS. We've got a blog post, we've got newsletters, we will be releasing an annual report soon, but it's also important to continue to give traditional species records to the recording societies as well. We'll be using those too. And then tell your friends about POMS, get people involved. We're on Twitter as well, I believe. So, and yes, using the hashtag FitCount. So, and anything you need to know, you can find on our website at ukpoms.org.uk. Thank you.